Thank you for attending today's virtual seminar titled A Story of Pill Stability, starring water activity, presented by Brady Carter.
I will be discussing today the idea of being able to utilize water activity in pharmaceutical products to try and look at pill stability, which is a big part of new ICH guidelines, quality by design factors, this idea of looking at what the stability of a pill is. And hopefully I'll be able to provide information today that will be helpful in being able to show how you might be able to take advantage of water activity in being able to determine the stability of a pill or a solid dosage tablet, a pharmaceutical product, and be able to control or extend that shelf life or that stability using these concepts of water activity. So what do you need to know to be able to determine the stability of a pill or of a pharmaceutical product? There are several different factors that work together in determining stability, but the main ones are moisture. Moisture is going to have an impact on what the stability of a pill. Most people intuitively will know that and will understand that moisture certainly plays a role, but may not have a real full idea of how to actually go about looking at that moisture. Temperature is another one that intuitively we would understand that as you increase temperature, all things start to speed up. So the other part of this stability is time, and how long will this product remain stable. Over time, because nothing here on Earth is in a true state of equilibrium, all things will change, and that's what we call shelf life. And so over time, the stability of a product will be reduced to a point at which that product is no longer deemed as being acceptable or that it can still be used. And there's going to be some type of product change that occurs during this process that's impacting or determining what that stability is. And it's important to understand what that product change is. It's not going to be the same for every type of pharmaceutical product. So there's usually typically some background work that has to be done to figure out exactly what product change it is that you're most interested in and in tracking that's going to determine what the actual stability of the product is. And of course, this is incorporated into new guidelines. Stability testing has been an important part of any kind of drug release program and required in order for that drug release program to be accepted. There has to be some way of showing that the stability of the product has been assessed and analyzed, and some estimation of what that stability is expected to be. And there are actually guidelines now with the introduction of ICH as part of a way of being able to globalize recommendations. In the ICH guidelines, they recommend for stability testing that you use 3 different temperatures, 25, 30, and 40, and then 3 different water activities. Now, these will be listed as relative humidities, and that's where there sometimes tends to be a disconnect in the area of pharmaceuticals, is an understanding that relative humidity and water activity are actually the same thing as long as you're in a sealed system, and that because they're the same thing, there are influences of relative humidity on the actual water activity of a product. If you're talking about, in general, if you're talking about the atmosphere around a material, that would be referred to as relative humidity, whereas what the water is actually doing in the product would be referred to as water activity. So when ICH guidelines outline to store at different relative humidities, what it actually means is that what you're trying to do is control the water activity of the product to those levels, and that water activity is going to then have some impact on the overall stability of the product. And so what we're going to focus on today is how do you track that moisture aspect of stability? Why do you control to different relative humidities, which in turn controls water activity? Yes, it does also control moisture content, but more specifically, relative humidity is directly associated with water activity. So should you be tracking moisture content when you're talking about product stability, pharmaceutical product stability, or should you be tracking water activity? And that's kind of the question we want to try and look at today. So let's talk a little bit about moisture content. Moisture content is a ratio of the amount of water or the quantity of water in comparison to either the dry weight of the material that you're dealing with or to the total sample mass. And that's typically referred to as either a dry basis or a wet basis reporting, which are not the same, by the way, and cannot be directly compared. So it is important to know how that calculation is actually made. But this is just a ratio of water to other materials. What is the quantity of water in comparison to what else is there? This is typically done either by what's called a loss on drying, where you weigh it wet, you dry it, and then you weigh it dry, and the difference between the two is the amount of water. Or by titration method, which is commonly referred to as a Karl-Fischer analysis, to determine how much water is present. Here's the problem with moisture content, though. These values that are obtained are all relative. There is no absolute value for water content. And that's because there's no way to know where zero is. There is no such thing as zero as a reference place for moisture content. So all values that are derived from moisture content are empirically derived because there is no way to be able to relate them to some standard value. There is no standard for moisture content analysis, even though there are standards programs where either a company sends out samples declaring them to be of a certain moisture content. In reality, that's not possible. They have to be still determined using some methodology, either titration or loss on drying. And so because of that, then all values are empirically derived, and there is no way of being able to figure out whether the value is correct or not, because it's impossible to figure out whether it's right if you don't know what the target is. So in terms of accuracy, moisture content values cannot be determined for their accuracy. Accuracy being whether you actually hit the bullseye, because without knowing where the bullseye is, it's impossible to know if you've actually hit it. So there's challenges associated with moisture content, even though from a concept standpoint, most people would understand moisture content and have used it, and it's a very typical way of looking at moisture in the pharmaceutical industry. Water activity, on the other hand, is a measure of the energy status of water in a system. As opposed to measuring how much is present, water activity actually tells you what the energy is in comparison to what it would be if you had pure water. So it's a ratio of how much energy is present, what's the total overall energy of water in a system in comparison to what it would be if you had pure water. And the way that that's measured is by looking at how many water molecules have the ability or enough energy to escape into the vapor phase and to exert a vapor pressure. And what that vapor pressure is in comparison to what the vapor pressure would be if you had pure water. And so water activity is specifically the water vapor pressure above a sample divided by the saturated vapor pressure at that same temperature. So if you have a product at a certain temperature and you can measure the vapor pressure above that sample and compare that to what the vapor pressure would be if you were dealing with pure water, you have water activity. So it takes on the form of a ratio from 0 to 1. The closer to 1, the more like pure water the water is in a product. And so the water activity of pure water, of course, is 1. And again, this is related to relative humidity. Instead of on a ratio scale, we're acting on a percentage scale, so 0 to 100%. So water, saturated water has, or a product at saturation has a relative humidity of 100% or a water activity of 1. And so we can look at this as being energy, and in fact, water activity is derived from second law of thermodynamics and through what we would call Gibbs free energy, where we can look at the amount of energy that's required to remove water is related to the gas constant multiplied by temperature, and then the natural log of this thing called water activity. So water activity is giving us a direct relationship to the energy that's required to remove the water. Hence, we say that water activity is a measure of the energy status of water. The more tightly bound, and you can see that I haven't used those words yet. The terms that often are associated with water activity are terms free and bound. Stay away from those, and I will stay away from them during this seminar because they mean different things to different people. especially in the pharmaceutical industry, where those terms tend to be very confusing, where people use bound water to mean several different things. In this case, when we say bound, we mean that the water, the energy of the water has been reduced, or that there's more work required in order to get that water out. And so hence the overall energy has been reduced of the water, and that's related to the water activity. That water doesn't have the ability to be able to escape into the vapor phase and contribute to a vapor pressure, so it's lower in comparison to the vapor pressure of pure water, and hence the water activity is lower. And that's also providing valuable information, as we will see, about other things that are happening in the product. So to visually show what's going on here, if we have a container and we were to place a pharmaceutical material in that container and then seal it up, over time, water would escape into the vapor phase, and the number of water molecules that escape into that phase and the pressure that's being exerted by those water molecules is going to be determined by what the overall energy of the water in that pharmaceutical product is. And so the more that water is interacting with things or being bound in that product through polar interactions, bonds we call ionic bonds or covalent bonds, hydrogen bonding, different interactions that can occur between water and other typically polar materials that restrict that water's ability to move into the vapor phase, will determine what the overall vapor pressure is. If we take that vapor pressure, or we are able to measure it in some way and determine that pressure, and then we compared that, as you can see in the container on the right, if we had pure water at that same temperature in a container and sealed up, it would exert a certain pressure, the number of water molecules in the headspace being dictated completely by the temperature. So if we know the temperature that the sample, the pharmaceutical product's at, then we can know what the saturated vapor pressure is of water. Take the vapor pressure over the sample and divide it by the saturated vapor pressure at that temperature, and you get water activity, assuming that that pressure and temperature are constant. And so it's important that temperature be controlled through this whole process, otherwise it invalidates the values. So from a practical standpoint, how is this done? How do you measure water activity? And it's actually a very simple measurement to do. The science behind it is very powerful, but as far as the action of taking a measurement, it's a very simple process in a benchtop instrument, as you see here. This is our Aqualab Series 4 TE instrument, and it works using what's called a chilled mirror or dew point method for measuring water activity. And how this works is that a sample is placed inside this chamber, it's sealed up. When it's sealed, water will start to escape into the vapor phase. Again, the vapor pressure or the number of water molecules in that vapor phase being dictated by the overall energy of the water. And now to figure out what that vapor pressure is, a mirror inside of the chamber is chilled or cooled down until dew forms, which then provides the dew point temperature. At the dew point temperature, the vapor pressure is equal to the saturated vapor pressure at that dew point temperature. So once that dew point temperature is determined, it can be used to determine what the vapor pressure is inside the chamber. Then if the sample's temperature is known or measured, which it's done through an infrared sensor, as long as that temperature is known of the sample, then the saturated vapor pressure can simply be calculated. So from the dew point temperature and the sample temperature, we can get the vapor pressure above the sample, the saturated vapor pressure, divide the two, and you get water activity. So this is a primary method of measuring water activity, by measuring the vapor pressure. There is no sensor that's involved to measure any kind of relative humidity, which means we don't have to wait for equilibration of the polymer in the sensor. Plus, we're accounting for any temperature differences by measuring the sample's temperature, which makes this whole process very fast, less than 5 minutes for most types of products, and very accurate, highest level of accuracy available in water activity testing at plus or minus 0.003. So it's a very reliable way of being able to determine water activity. Very simple benchtop instrument. You put the value or the sample in, you close the lid, water activity runs, it finishes, and the value is reported onto the screen. And this instrument has the capability of being able to interact with a computer program, which will record the data as well. And it also comes complete with Part 11 compliance, administrator controls, password protected. All of those are incorporated into the instrument. So what are some water activities of common pharmaceutical types of products? There is a range represented by different types of products, anywhere from a compressed tablet, typically around 0.39. Some can be lower than that, depending on excipients that are used and how they're processed. Liquid fill capsules at a similar value. Powdered capsules are a little bit lower water activity. Again, all depends on what type of, how the product is manufactured and what's used. Some water activities of different types of potential excipients are shown there, or additives that may be used. And then you have oral liquid suspensions that are going to be at a high water activity, a high level of water present, so their water activities are quite a bit higher. So a range of water activities are represented by pharmaceutical products, and depending on what that range dictates what's going to be important in terms of the stability of that product, as we will see. Now, there is a relationship between water activity and moisture content, and that's referred to as the moisture absorption isotherm. Moisture absorption isotherm, meaning at one temperature, the relationship is specific to the temperature. And notice that from this curve, and this is just a generalized form, actually every material will have a very unique shape to its isotherm, and the isotherm provides information about how a product is going to take up moisture as its water is introduced to the product or lost from the product. So you have both adsorption, taking up moisture, and desorption, losing moisture. It also provides valuable information about the nature of the material, how hygroscopic it is, how it will react if it's combined with other ingredients. All this information is provided by this moisture absorption isotherm. And I'll reference to this several times as I go through the rest of this talk. But it's important to have this understanding of this relationship between water activity and moisture content. Again, water activity being the energy, moisture content being the quantity of water. And how do you measure moisture absorption isotherms? So we're finding two things that we need to know from this. We're going to have to be able to measure water activity, but in order to know what water activity is important for stability, we're going to need the moisture absorption isotherm. We can measure water activity using our Aqualab Series 4 instruments, benchtop instruments. But how do you actually measure this moisture absorption isotherm, or this relationship between water activity and moisture content? And that's done using a moisture absorption isotherm generator, which our version that we sell from Decagon is called the Aqualab Vapor Absorption Analyzer. This is a very simple to use instrument. It is a higher sample size type of an instrument, so this would be primarily used for finished product, not necessarily for active ingredients. Sample size is typically 500 milligrams and up. It has the ability to be able to do both what's called static or DVS tests, where you hold at certain water activities and watch for equilibrium by weight change, or by tracking the weight change. But it's unique from all other types of instruments that do this work in that it can also do what's called dynamic isotherms, where instead of controlling to a specific water activity, the product is exposed to saturated or desiccated conditions, and then the changes that occur are tracked dynamically, making this a very fast process with very high data resolution. And I'll show later on where the times when you would use the static and the times when you would want to use the dynamic. And there are reasons to have both, and this is the only instrument, the Aqualab VSA, or Vapor Absorption Analyzer, is the only instrument that has the ability to do both types of isotherms. So now let's get into talking about specifically drug stability and water activity. How are these related? What are the applications that water activity is involved with? And how can it be beneficial and helpful in being able to determine the overall stability of a drug product? And first we're going to talk about microbial growth. Water activity, if it's known for anything, it's known for its ability to control microbial growth. And this has been known since work done in the 50s. And in fact, in an indirect way, it's actually incorporated already into ICH guidelines. Although in these guidelines, what they say is there evidence, scientific evidence, that a product is inherently dry or dry, so dry that it will not support the growth of certain microorganisms or of microorganisms in general? And if so, then microbial limits acceptance testing may not be necessary. And this is built right into decision trees within the ICH guidelines. However, there is no clear indication in these guidelines what it means to be dry, or how you actually would track whether or not a product is inherently dry. The intuitive response is, well, they mean moisture content, and so we run Carl Fisher on those types of products. But in reality, moisture content has no relationship whatsoever with microbial growth. Water activity is what's actually determining whether microorganisms grow. A microorganism doesn't care how much water is present. It cares what the energy of that water is and whether it can access that water or not. And so in terms of these decision trees, when it says, is a product dry, and is that inherent dryness proof, is there scientific proof that it won't support growth? The only way you can do that is through water activity and not through moisture content. So how do you determine if a product is dry? Again, you have to go to water activities water activity. In terms of microbial stability, that's the only way to do it. And that's even verified through USP chapter 1112, an informational chapter that talks about this idea of water activity being what controls microbial growth. And there being justification for eliminating microbial limits testing if you can show that intrinsically your product is dry because it has a low water activity and will not support growth. That can't be done with moisture content. Again, microorganisms don't care how much water is present. They only care if it can access that water. What's the energy? And this was seen, again, since the 50s, work done by Scott, where he saw there was no correlation between moisture content and microbial growth, and instead established growth limits in terms of water activity for different microorganisms. And you can see what those different limits are. They're shown in this table for the gram-negative, gram-positive. Each type of organism, even within these groups, each specific species of organism has a specific water activity level at which it stops growing. And this is all related to the ability of each organism to react to what we call osmotic stress, where it's been exposed to an environment where the energy of the water is lower than the energy of the water in the organism itself. And it relies on being able to transport by energy ratios water from its surroundings into the organism in order to access it. And so if it experiences conditions where the water activity of the environment is lower than the water activity inside the microorganism, it experiences what's called osmotic stress. And each organism has a unique ability to react to that and respond and adapt. And so the limit where they stop growing is dictated by what adaptation abilities that microorganism has. But each one will have a limit where below that water activity, this organism will not grow. Now, this is not a sterility step. It's not a kill step. It will not remove all organisms. It simply prevents them from growing or proliferating to values that could then cause infections later on. So this is a preventative step. Once you've gone through your good manufacturing practices and created a product that has a low water activity, as long as you monitor that water activity and you're assured that there is no external source of an inoculant, then that product will remain safe. And you can see that all microorganisms stop growing at 0.6. So when you start talking about solid dosage products or capsules, these all inherently have very low water activity down, as you saw, in the 0.4 range. So there simply would be no reason to do extensive microbial limits testing on these products as long as you can assure that they haven't been inoculated by an outside source or by the manufacturing process. Now, moving on from microbial stability to other things that water activity influences, water activity also has a large influence on the reaction rates of chemical reactions. And these are going to be ones that are important for the stability of the active pharmaceutical ingredient and will influence the degradation of the active pharmaceutical ingredient. So if you want to talk about API degradation, then you do have to talk about water activity because it will influence that rate. The kinetics of these reactions that influence this degradation can be determined and can be related to both water activity and temperature, but they both have to be accounted for. And typically when this is done through something like accelerated stability testing, often the mistake is to only account for the temperature effect, and that's typically done through an Arrhenius type of relationship where you model the effect of the temperature by collecting information or data at several different temperatures. But it's important to understand that both temperature and water activity have their own individual impacts on those reaction rates. And if you only account for temperature, then the active ingredient may be degraded due to high moisture or changes in water activity rather than changes in temperature, and it won't be possible to account for that if that wasn't taken or included as part of the modeling that was done for looking at these chemical degradations of the active ingredient. So in general, as water activity increases, so do the reaction rates of the several different types of reactions, and be hydrolysis reactions, enzymatic reactions that could potentially break down an active ingredient. All of these reactions are going to increase with an increase in water activity. The rate at which that increases, again, is specific to the material itself and the reaction, but it can be modeled using some experimental work where you look at several different temperatures and water activities and track the progress of a reaction, combine that information, and using QA or Q10 values and a combination of those, it's possible to actually model the kinetics of those reactions according to the effects of temperature and water activity. But water activity does have to be part of it. Water activity certainly has an influence on the integrity of gel coatings. If you're looking at gel capsules or gelatin-covered liquid capsules, the integrity of those capsules and whether they become sticky or not, which is a common occurrence, and then weak, and they tend to dimple, or you might see cracking if the water activity goes too low. This is all, again, influenced by what the water activity is, and that will then be influenced by both how the product's manufactured, what's the water activity of the ingredients inside of it, and what kind of environment is it exposed to, and how good is the packaging material in keeping it from being able to, or going to higher water activities. And so you can see here there's a couple of videos that are very interesting to look at. The first video will show the nature of liquid gel caps when they are at essentially room humidity. So you can see as this video progresses, you can see what the water activity is on the screen of that product. It's at about room humidity, 34% relative humidity, so 0.34 water activity. Notice as the cup is as the cup was shaken, those pills remained free-flowing. They weren't sticking together. They were loose and could be and were able to shake and move them. Now in the second video, now you can see that the water activity of the gel caps has been increased. It's been exposed to 75% relative humidity, meaning its water activity has increased to 0.75. You can see at this water activity, as the cup is shaken, those capsules stick together, and that's because of exposure to high water activity. So it very clearly shows what the effect of an increase of moisture, and again, this is all done at room temperature, no temperature effect. This is the effect of water activity of the capsule coating, causing it to become sticky, and then those capsules will stick together due to that exposure. So it's a great example of how water activity influences the integrity of these different types of products. And so maintaining or controlling that water activity, and that's ultimately what packaging is doing with a high moisture barrier, is controlling the water activity. It's directly controlling that, but throughout, or the common practice is to try and look at that in terms of moisture content. Well, it's indirectly controlling moisture content, but what it's directly controlling is, again, the water activity. What about moisture migration in capsules? This tends to be a big problem and a ubiquitous problem in pharmaceuticals, especially when you're creating capsules where you're putting some sort of an active ingredient and an excipient inside of some sort of a capsule. If water activity, or if the water activities are different between the capsule and the material that's inside, then you will experience moisture migration because water moves from areas of high water activity to areas of low water activity, not from high concentration to low concentration. And this is critical. You cannot prevent moisture migration by matching moisture contents because at a given moisture content, the capsule and the ingredients inside will not be at the same water activity. They have to have their water activities matched in order to prevent moisture migration from happening. And so if you want to create some sort of a capsule with a medicine inside of it that is going to be stable and not experience any kind of moisture migration, then you have to manufacture them so that they have the same water activity and check what those water activities are. And then if they're at the same water activity, then they'll remain stable and there won't be any moisture migration that could cause either the active ingredient to degrade at a higher rate based on what we just saw, water activity's relationship to chemical reactions, or cause the capsule to dimple or to crack due to moisture either entering the capsule or being removed from the capsule, depending on what the ratio is. And so this is an example This is a little experiment that we ran to show how this actually works, and the fact that if you do put them together at different water activities, you can actually model what's going to happen due to this moisture migration that might occur. So the question here is, what would happen if you were to put MCC inside of a capsule, and the MCC is at a very high water activity at 0.73, whereas the capsule is at a very low water activity at 0.43. And these ratios are quite large, and that was, of course, to be able to show and clearly see what's going to happen. But in reality, those ratios would likely not be that large. But in a similar way, if you were to put them together at different water activities, they are going to exchange moisture and come to some equilibrium water activity. What will that water activity be, though? Where will they come to equilibrium? The answer is not the midpoint or the average between the initial water activities of the two ingredients, because it's dependent upon their moisture absorption properties of each of the individual components, the capsule as well as the ingredient inside. So the way that we do this is that we model it by doing what's called a combined isotherm method, where we can look at the combined moisture content of the ingredients, and then using modeling techniques of the individual isotherms for each of the ingredients, we can create what's called a combined isotherm, or the coefficients for an isotherm that combines the properties of the two different isotherms, and then use the information from that to figure out what the equilibrium water activity is. And from that equilibrium water activity, we can then apply that to the model for each of the individual isotherms to figure out what, or predict what, their moisture content will be when they get to that equilibrium water activity. So this will say, if I were to put those two materials together at different water activities, where will their equilibrium water activity be, and what will be the moisture content at that equilibrium water activity? And this is all done, again, modeled without actually having to create the product, so that you could get an idea of what is actually going to happen if I were to combine two ingredients at different water activities, where will they come to equilibrium, and what is the consequence going to be at that equilibrium? Will that cause an unacceptable level of degradation to the active ingredient, or will it cause some sort of a deformation in the capsule or the coating of the material or the pharmaceutical product? This modeling that's done, I'm showing here, we're not going through step by step, but certainly we have the tools to be able to do that. And if you're interested in being able to do this work and this modeling, as well as many of the other things that are shown in this seminar, I do offer this in an Excel form or an Excel sheet where you can put some information in, and then the calculations are done automatically. We are in development of a software tool that will actually do all of this as well in a much easier to use non-Excel format that will become forthcoming soon. Again, it's in development right now. So let's take a look at what happened. We did these experiments. We combined, again, 0.73 MCC into two different types of capsules, the first one being made of HPMC capsule. So we put in MCC. You can see there to the right what the isotherms look like for each of those individual products. So we used the combined isotherm technique that I just showed in the previous slide to do a prediction or determine or predict what we think the final water activity would be based upon the individual properties. And then we actually combined them together and let them sit for an extended amount of time and come to equilibrium. And then we measured what the actual water activity was to see how well our prediction or how good a job of predicting we could do. So here we have a situation where you've got the initial water activity of the powder is 0.73, and it's at about 8.95% moisture. The initial water activity of the capsule is 0.439, and it has a moisture content of about 5.6%. So what happened when we combined them? We predicted that it would be a final water activity of 0.657, and in reality we got 0.551. So not exactly the same, but a very good prediction of the range or area where that water activity would be, which then could be considered for, is that going to be okay from a microbial standpoint? Is that going to be okay from a stability standpoint if this actually was happened and these were combined? We predicted the final moisture content to be 8.33% for the MCC powder. It was actually 6.7% at that lower water activity. And we predicted that the moisture content of the capsule would change from 5.5 to 8%, and in reality it was 6.2%. So the predictions weren't exactly right, but they were close and gave us an idea of what was actually going to happen with this product. We did this also for a gelatin-based capsule where the powder at the same conditions, 0.73 water activity and 9% moisture. But in this case, we're added to a gelatin capsule, who had a water activity of 0.43, but a moisture content of 15%. Now, if you were to look at this example and intuitively thought that moisture content controlled this, the idea would be that moisture would move from the capsule into the powder based upon just looking at moisture content. That can't happen because it's at a lower water activity than the powder is. And so you would expect, if that were the case, that when they come to equilibrium, the moisture content of the capsule should have decreased if moisture content was actually controlling it. Well, let's look at what actually happened. So we predicted the water activity of the powder to be at 0.683, or both of them to come to equilibrium at 0.683. In reality, they came to equilibrium at about 0.62 water activity. So in this case, the prediction was actually very good, very close to the actual water activity. We predicted the final moisture content of the powder to be 8.5. It was actually 7.1. And we predicted the final moisture of the capsule to be 17.8. In reality, it was 17.3. So a very good prediction of that final moisture content. Notice that the moisture content of the capsule did not decrease, and in fact, it increased as a result of these being placed together. So if you were to try to go off moisture content, this would not work at all, because it's not quantity that controls migration. It's going from high energy to low energy that controls that moisture migration. Okay, so we've talked about how water activity influences microbial growth. We've talked about how it influences coating or capsule integrity. We've talked about moisture migration within capsules that affects its stability. Now let's talk about excipient selection and how water activity plays a role in terms of determining the overall stability, since the excipient, being the largest concentration ingredient, has a huge impact on the overall stability of the product and the stability of the active ingredient. And so when determining excipients, there are several different factors influenced by moisture that should be considered when looking at an excipient. One is how hygroscopic it is. One is its glass transition properties, its deliquescence properties if it's crystalline, and absorption kinetics, or how quickly does the moisture change in the product. So if you were to do this and consider different types of excipients, one of the first things you want to do is look at how they respond to moisture. Are they hygroscopic? How hygroscopic are they in comparison to other types of excipients? And so on this graph, you'll see several different types of excipients. There's MCC shown, carmellose sodium, sucrose, sorbitol, and mannitol are shown here. And each of them have very unique, as you'll see, or as you can see, very unique moisture absorption properties. Some of them are quite hygroscopic, and how hygroscopic they are is dependent upon the slope of the isotherm curve. The steeper the slope, the more hygroscopic the material is. It's taking up more moisture without a subsequent big change in water activity. For isotherms that are very flat, and this is typical, as you can see, of the crystalline types of materials, that very flat isotherm is typical of crystalline because the crystalline lattice won't let moisture penetrate in. And so any water that comes in causes big changes in water activity, but very small changes in moisture content until it reaches its deliquescence point. And you can see that there are several that do go through deliquescence and then become extremely hygroscopic and go into a saturated slurry form. So depending on which water activity you're targeting and which types of properties you want the material to have in terms of its absorption properties would influence which of these excipients you might choose for your particular product. This is a very fast way to do a relative comparison of both how hygroscopic they are as well as their maximum water absorption at a given water activity. And that's just simply you compare where the curve is in comparison to the other two. And notice that those values change depending on where you are because of the crystal materials experiencing deliquescence. So a very good, very fast way. These are dynamic curves. I mentioned earlier the ability of the VSA to be able to do both dynamic and static. These are dynamic curves. They're very fast. Each of one of these was done in a matter of a day as opposed to the 2 weeks it can take to do a static isotherm. So very fast for a very quick comparison to determine how hygroscopic they are. They aren't necessarily at full equilibrium in each of these curves, but it's certainly possible, since they were all treated the same, to get a relative idea of how they respond to increases in moisture. How about glass transition? Glass transition, of course, has a huge influence on excipients that are in an amorphous state. If they go through glass transition, all things start to speed up once the glass transition has occurred for those materials that are in the glassy amorphous state, which is a metastable condition. And so the excipients that are in that state are offering a stability to the product by being in that condition. But if they experience a glass transition, which can be induced both by temperature, what's often referred to as the glass transition temperature or TG, or by moisture can also induce a glass transition because it increases plasticizer and can cause it to get to a critical point where it then moves from glassy to rubbery. Once that happens, everything speeds up. Reactions that degrade active ingredients, dissolution properties change, caking and clumping starts to happen, all as a consequence of having experienced this glass transition. So it's important to have an understanding of what those properties are for different types of excipients, and when will temperature do it, but also when will moisture do it? And that part's often overlooked in terms of the glass transition. And as you can see on this curve, again, this is a dynamic curve, high resolution. Because of the high resolution, we actually can see these inflection points in the absorption curve where the absorption properties change drastically. And that first inflection point that you see in the curve actually is the critical water activity for glass transition, equivalent to what you would call a TG. But in this case, it's in terms of water activity instead of in terms of temperature, because we're scanning water activity. And inducing this change, this inflection point represents the point at which it transfers or transitions in the time of the isotherm from the glassy state to the rubbery state. So if you were to move the water activity to a value higher than that for a given excipient, it will experience glass transition, and then its stability, of course, will be drastically reduced. So this critical water activity, and it will go down as temperature increases, so it is influenced by temperature. So simply by running these isotherms at multiple temperatures, you can gain an idea of what water activity you have to be able to maintain at any given temperature in order to prevent glass transition from happening. So that becomes a critical value. You manufacture to water activities lower than that with a safety factor built in, and then you use packaging that's going to prevent it, no matter what conditions it encounters, from going above that critical water activity, and that will maintain the stability. So if you were to compare two different excipients, this is a great way by comparing their dynamic isotherms, which one has the lower or the higher critical water activity would have an influence on what one you might use for a particular excipient for a different product as you establish a drug release program. With the system, the VSA system, you can actually see glass transition occur with these dynamic curves and then switch to the static, which then induces crystallization when you move to the static, which is characterized by a distinct drop in the isotherm curve. As the crystal lattice forms, it pushes water out. That water escapes and causes the moisture content to decrease at a given water activity. Very unusual, but typical of crystallization. With the utility of our vapor absorption analyzer, you can actually use the system to see this on one sample. You can induce a glass transition and then watch it crystallize as a consequence of the glass transition. This is the only system that allows you to be able to do that. And it's also very easy with these dynamic isotherms to be able to observe deliquescence. Deliquescence, of course, is the crystal lattice going into a saturated state due to encountering a specific water activity, or the deliquescence water activity. And that happens at a very specific water activity depending on what the material is, as well as the form of the crystalline material. And if you combine crystalline materials together, then it's the product of their deliquescence points determines where they actually will go through deliquescence. But as you can see from this dynamic curve, it's very clear it's a 90-degree turn where that happens. Obviously, if you move past that deliquescence point, then stability is going to be gone for that product because it's going to start to go into saturation. All reaction rates are going to drastically increase. If you can maintain below that deliquescence point, there are possibilities for just powders or crystalline powders to become sticky at values less than the deliquescence point, but it won't have the huge impact on the dissolution properties of the material. So having an idea of where that deliquescence point is critical in comparing different excipients. And then finally, what are the kinetics absorption of the different types of excipients? This is in reference to the diffusion properties of the material. How quickly does water diffuse through those different materials that would then cause or result in a change in water activity when exposed to higher conditions. And so you can determine using information collected from static isotherms, and that's what you're seeing here is what we would call a kinetic curve. You're stepping to different water activities, and then the weight changes, and then flattens out, and when it flattens out to a certain rate, you would deem it as being equilibrated. If you track the time that it takes from when it starts to when it ends, and you can use that information to determine a time constant, and then you can compare time constants at given water activities for different excipients and find the one that takes the longest, so that it would be the slowest to respond to exposure to different conditions or high relative humidities, which then in turn would help to maintain its stability. And so this information can all be gleaned from data taken from, again, our vapor absorption analyzer that I showed earlier, has the ability to be able to collect this information, and then the calculations can be done to figure out what those what the diffusion coefficients or diffusion properties are of a particular material. Now let's look at what influence the water activity of the excipients can have on tablet hardness. So water activity also has an influence on textural properties of the tablet material and how well they compact, and what the hardness is that results from that compaction process. And the idea here is that there likely is going to be a water activity that's the ideal value. And if you get to too low values or too high values, that will cause those the excipients to not perform as well when they're being compacted into a tablet form. And so we put this to the test. There's 3 different excipients shown here that were controlled to several different water activities, made into tablets, and then tested for tablet hardness using a texture analyzer. And you can see reported here is what we're calling the break force, the force in grams of where the tablet that was formed broke. And you can see that there is that trend that we would expect for at least 2 of the excipients, the MCC and the sorbitol, where they do peak at a certain water activity and have their maximum break force, but then if you go to higher water activities, then they actually start to lose break force or become softer. And then at lower water activities, they also are softer. So there does appear to be an ideal water activity range. It may not necessarily be at 50. The peak could actually happen somewhere between 30 and 50% relative humidity, but somewhere in that range, there is an indication that there is a peak point for performance of those the tablets that are made from these excipients. So with some additional investigation, it is possible that you could identify the ideal range of water activity, where if you're too low, that's not going to be good, or if you're too high, it's not going to be good. And so you would want to make sure that your excipients, before going into the tablet press, are controlled or conditioned to that given water activity. And again, you could check that very easily through the use of a water activity instrument. What about tablets and where they're stored, or the type of humidity they're exposed to, and which would then cause their water activity to change? Is there a similar type of effect? And you can see here, these are several different types of tablets that were controlled to different water activities and then again tested for tablet hardness. You can see there are some that in general, as you increase the water activity, the hardness decreases. There are types that have the peak that we saw with the excipients. There is a water activity where it's at its maximum hardness or the maximum level of break force required, but then it decreases at higher water activities, but is also lower at lower water activities. And then some that don't have much of a response at all to increases in water activity. So it would be important from this standpoint to establish for a given product that you might manufacture, where is that ideal water activity range, or how much does water activity influence the hardness of the tablets if it's exposed to different conditions over time. Where's that ideal range? Make sure that it's manufactured to that water activity by checking with a water activity instrument, and then packaging such that if it's exposed to different or higher humidity conditions, that it won't get to water activities where those properties could be lost, which of course will then impact dissolution. So how do you actually track these changes that might occur in packaging? And you do that, of course, through package permeability. And you can determine what the package permeability is. It's typically reported for different products, determined using ASTM standards that provide what's called a water vapor transmission rate. And then you can take that value and plug it into equations that are based on Fick's first law of diffusion, and figure out what exactly how much will the water activity of the product in the package change based upon its permeability properties, as well as the conditions that it's exposed to, and by establishing some sort of a critical water activity based on the information that we've already talked about. So all the work prior was to figure out where is that critical water activity that's going to maintain the stability for the amount of time that's acceptable. Once you've decided or figured out what that is, and you know the water vapor transmission rate, you do have to convert that into terms that can be used, permeance being the one that would be able to be plugged into Fick's first law. You can't plug water vapor transmission rates into those models because they aren't derived from first principles and they don't account for the differences in vapor pressure inside and outside the film. So you do have to know the conditions under which the water vapor transmission rate was determined. If you do know that vapor pressure difference, you can then convert that into a permeance value, which you can then plug into Fick's first law and do calculations using those modeling techniques to figure out how much will the water activity of a product change based on the conditions it's exposed to, what kind of packaging you're going to have, or what kind of permeability properties will it have to have in order to achieve a desired shelf life under specific conditions, or what the actual shelf life will be given a permeability of a packaging and the conditions that it might be exposed to. And again, these calculations we have available through our Excel sheet that we're happy to provide in order to be able to do these calculations on your own for your particular product. And all of this is associated with if you want to, as part of a drug release program, do what's called accelerated stability testing, which is kind of a new buzzword in the pharmaceutical industry, be able to establish what the stability is without having to wait for an entire year to do real-time stability testing, then you can use what's called accelerated stability testing. This has been done for many years in the food industry, called accelerated shelf life testing. But the idea here is that you increase the conditions that a product's exposed to, trying to speed up all the processes that could happen, the degradative reactions, and then try to model that and then extrapolate back to what it would be under normal conditions. In this process, the important thing, and what I would hope is the take-home message from this, is that if you do that and you only account for temperature, you're only getting half the story. You have to account for temperature and water activity, and moisture content isn't going to provide the information that you need or give you the correlations that you need to what's actually happening in terms of stability. You get your temperatures, you get your water activity, you track the progress of whatever it is that's degradative, and then you can combine those and do modeling. Again, the ability to do that built into our Excel sheet, but it has to include both temperature and water activity to be able to do that correctly. And ICH shows that. That's the reason why they have a suggestion for temperatures, but they also have a suggestion for relative humidity. If they have a suggestion for both, it would infer that both have an influence on the stability. If you do the work and you only model for temperature, then it eliminates the whole purpose of doing it or considering accelerated humidity conditions. And the reason why that influences it is because it influences the water activity. And we've seen from this discussion today that water activity then influences many of the different factors that impact stability, from microbial growth to active ingredient degradation to gel coating to capsule moisture migration, selecting the appropriate excipients, the hardness of tablets and their dissolution, and to the packaging that's used in being able to prevent changes in water activity. So water activity overall is the best way to be able to monitor moisture and the effects of moisture on stability in pharmaceutical products. It's a very easy test to run on a benchtop instrument, and so it becomes very effective as a control parameter for stability as you create your drug release and stability program. It's our hope that you will include and consider water activity, and if you have questions on how to most effectively do that, I would certainly encourage you to contact us or contact me. Here at Decagon, we're more than happy to give any kind of application help that we can, as well as we pride ourselves on being able to know and provide the science that goes behind water activity, which then impacts all that you might do or potentially be able to do with water activity in your product. And with that, I thank you, and that will finish our seminar.